Dear all, today I'm going to tell you about titration of amino acids. Amino acids in water are ionized and act as either acid or base. You can see this is base and this is acidic form, and it ionizes in presence of water. This property is important to understand many properties of proteins and in protein sequencing, separation, identification, quantitation, and quantitation of proteins and quantitation of different amino acids, which are dependent on the characteristic acid base property. All amino acids having single COH and COH and single NH2 group crystallize to form neutral aqueous solution in a fully ionized species, which is known as zwitterion or dipolar ion. They are electrically neutral, of course, and don't move in an electric field. These cannot move in any electric field because they have equal positive and negative charge, which makes them neutral. Now, how can they be titrated then? Since this molecule can act both as base and acid or proton acceptor and donator, it can donate proton, it can accept proton. So such a uh, compound is also known as ampholyte. These are also known as ampholytes, and a simple monocarboxylic acid. Monocarboxylic acid is actually diprotic acid. This is zwitterion, दो ions है. This is an ampholyte because it has acid and base character. Plus, it is actually diprotic acid when it is fully protonated. It can be protonated, which means this will be there with positive charge, and if we add protons here, so this will be fully protonated and will be called as diprotic acid. Now, in this form, since I'm discussing the titration of these ions, so what will happen when we add NaOH to it? Here, whatever I have already told you, you can see this here. This is an zwitterion. If we reduce pH, if it is 7, then below 7, 1 to 7, protons will be added to it, as a result of which a proton will add here because this is fully protonated, and this will add here, and net charge on this species will be positive. If we increase pH, then what will happen? This addition of this will lose proton. On addition of OH, this will lose proton, and this species, this NH3, will lose protons, as a result of which net charge on this molecule will be negative. So if it is a Zwitterion can exist at IPH. IPH is known as isoelectric pH, and at this pH, this molecule remains neutral in charge because these two balance each other, and this species will not migrate to either anode or cathode when placed in an electric field. And this Zwitterion is now accepting proton, and this species forms. When it loses proton, this species form. This is acid acceptor, and this is uh, proton donator. Dissociation constant of amino acids can be determined, for example, by titration of acid. Titration, you already know that in acid, some molars of alkali is added, and as a result of which water molecules are formed, and this is how acid is titrated with the help of alkali, and various molarities and pH and various properties of that acid can be found out by the this experiment of doing by doing titration. Now, pKa for the acid, when a weak acid, for example Ha, is titrated with an alkali, then at the point of half neutralization of Ha, the titration curve shows an inflection and pH of the solution equals pKa of the acid. pKa is pH at which acid is half neutralized by addition of alkali. And what is inflection point? You can show this region. This is inflection point here, and this is inflection point here. At these 2 points, acids are half neutralized. Here they are not neutralized, they are fully acidic in nature. Here half of the acid is neutralized by the addition of OH, and then finally it is completely completely titrated, and all the it loses, actually this compound loses all its protons because of the addition of NaOH. So this dissociation constant for the acid, if it is weak, can be calculated, and addition of alkali is required, and Ha can be calculated, and pH of the solution equals the pKa of the acid. Since an amino acid has at least two acidic groups, at extremely low pH, its titration curve shows two inflection points with separate pK value for carboxylic group and amino group. I told you about the protic acid. So, Zwitterion uh, of amino acid could be converted into diprotic acid by addition of, or by lowering, addition of proton, or by lowering the pH of the medium. And when it is fully carrying, totally protonated, it is known as diprotic acid. And then from here, this point, uh, we can adding alkali, and find out the course of titration. And to find out the pi, this pi money, isoelectric point, can be approximately as halfway between the 2 points of the strongest buffering capacity, and can be estimated by this formula, which is pi is equal to half pk1 plus pk2. where Ki and K2, K1 and K2 are dissociation constants for the deprotonation of glycine. If the amino acid glycine is used, uh, glycine's carboxylic acid and amino group as shown in the next graph. Here, 0.1 molar and NaOH equivalents added, and plot has two distinct stages, each corresponding to removal of one proton. This is one stage and this is second stage. Here the removal of proton is taking place from COOH because this is more acidic. So first the removal of proton will take place from the acidic group, and this will result in total removal of proton, and the pH at this point will be IPH, isoelectric pH. Each of the two stages resemble in shape. You can see these will resemble in shape the titration curve of a monoprotic acid. अगर आप एसिटिक एसिड को टाइट्रेट करें तो आपको वहां पे एक ही प्रोटॉन है CH3COOH, जिसका सिंगल COOH एक ही है, तो एक प्रोटॉन का टाइट्रेशन होगा और इतना हाफ हाफ कर्व आएगा और उसकी शेप भी ऐसी ही आएगी. अब यहाँ पे दो प्रोटॉन्स हैं, यहाँ पे भी प्रोटॉन है, यहाँ पे भी प्रोटॉन है, तो सेकेंड स्टेज में फिर वैसे ही चूंकि 1/1 प्रोटॉन का टाइट्रेशन हो रहा है. सो हियर ये जो ये शेप है आधी शेप, और इतनी जो आधी शेप है, ये दोनों अलग अलग सिंगल प्रोटॉन के टाइट्रेशन को दिखाते हैं. नाउ मोर एक्सप्लेनेशन for this titration curve. First stage, at the beginning of titration, that is from here, if it is glycine, then predominant species at this point is a diprotic acid NH3 plus CH2 COOH. At midpoint of the titration, half the COO group has been already titrated. So क्या होगा, दो स्पीशीज इस पे यहाँ पे रहेंगी, एक NH3 CH2 COOH एंड दूसरी NH3 CH2 COO माइनस. दोनों स्पीशीज रहेंगी. अगर ये स्पीशीज है दिस इज प्रोटॉन डोनर, और अगर ये स्पीशीज है तो दैट इज प्रोटॉन एक्सेप्टर. तो पीके पर ये जो पीके पॉइंट दिखाया जा रहा है, इस पे ये अगर ये 10 मॉलिक्यूल्स हैं तो इसके 5 मॉलिक्यूल्स यहाँ आते आते टाइट्रेट हो गए, तो टाइट्रेट होके इस फॉर्म में बदल गए. सो हाफ 5 मॉलिक्यूल्स इस फॉर्म में है और 5 मॉलिक्यूल्स इस फॉर्म में है. ये मॉलिक्यूल प्रोटॉन डोनर है और ये मॉलिक्यूल प्रोटॉन एक्सेप्टर है. And pH at this point, pH at this point is equal to pK of the protonated group being titrated. इस पॉइंट को, इस pH को pK कहेंगे, डिसोसिएशन कांस्टेंट. पे क्या pH है वो इस ग्रुप का टाइट्रेशन पॉइंट होएगा. नेक्स्ट इंपॉर्टेंट पॉइंट कहाँ है, इस पे है. when all COH have lost their protons, अब कौन सी स्पीशीज डोमिनेट कर रही है, या वो खाली वही प्रेजेंट है, तो वो स्पीशीज है ये, जबकि this has lost all its protons. That is why this is there. And उस पॉइंट पे जितने भी OH molecules add हो चुके हैं. This pH at this point is known as isoelectric pH. Now, removal of H from NH3. So, this is the second stage of titration. At midpoint, again, equimolar concentration दो स्पीशीज की दिखेगी. कौन सी? यहां से टाइट्रेशन अब सेकेंड स्टेज में शुरू हुआ है. तो again, if it is 10 starting point पे, this was 10 in number and this is again 10 in number. तो halfway पे इसका हाफ टाइट्रेट हो चुका है, यानी कि 5 मॉलिक्यूल्स इस स्पीशीज के हैं और 5 मॉलिक्यूल्स इस स्पीशीज के हैं. Equimolar quantities are present. This is pK2. pK2 यानी कि pH at which this both species are present in equal concentrations. और जब completion हो गया titration का, now no proton is left with the molecule. तो यहाँ पे भी कोई प्रोटॉन नहीं है, यहाँ पे भी कोई प्रोटॉन नहीं. This means titration is complete. Now, what information do we get from titration curve? क्यों ये important है, ये हम आगे देखेंगे. First of all, जो हमने अभी आपको बताया था, this is again depicted here. यहाँ पे pK1 है और ये pK2 है. और जितने भी अमीनो एसिड्स हैं which have one carboxylic group and one amino group, they have approximately they have same shape of the titration curve. और उनका जो pK1 है, pK2 है, IPH है, वो भी almost एक सा होता है. Now, ये whatever I already explained you, that is written here. And now next slide. Amino acids like glycine, glutamic acid, histidine, in which R groups are charged, having acetate or basic side chains, giving them more than 2 dissociable hydrogen ions. Lysine में 3 बेसिक अमीनो एसिड है. इसमें 2 अमीन्स हैं और एक कार्बोक्सिलिक ग्रुप है, all of which could be fully protonated. So they can lose 3 times hydrogens, protons in when they are titrated. Here glutamic acid में क्या है? It has 2 carboxylic group and 1 amino group. Again it has 3 groups which can lose their proton. And histidine is also like that its R group is charged and it can lose proton. Generally pK1 अब now denote कैसे करते हैं? तो pK1 is for carboxylic group, pK2 is for amino group and pKr is for ionizable group in the R chain. Side chain में जो ionizable group है उसको R group से denote करते हैं. Charged amino acids possess R groups. For example, glutamic acid has carboxylic acid side chain in addition to its alpha carboxylic, जो मैं already बता चुकी हूँ. तो curve will be more complex. 3 parts आने चाहिए इसके. 3 times एक group 1 by 1 titrate हो रहा है. Titrate क्या हो रहा है? Proton is binding with OH group and there are 3 groups carrying protons. So 3 times 1 by 1 they will be titrated. Glycine to be glutamic acid में अब now from this or this graph you can see that pK2 is in 9.6 that is a basic range, while this is an acidic range. Now, what will happen if there are three titratable groups present in amino acid? तो ग्लूटामिक एसिड में दो कार्बोक्सिलिक ग्रुप्स हैं. pK1 alpha carboxylic group will be for glutamic acid is 2.2. 2.2. pK2 is gamma carboxylic group 4.3. दोनों जो है एसिडिक ग्रुप में होंगे, एसिडिक पीएच के रेंज में होंगे क्योंकि कार्बोक्सिलिक ग्रुप्स हैं. देन वन इन बेसिक रीजन, क्योंकि अमाइनो ग्रुप के टाइट्रेट होने से, NH3 ग्रुप के टाइट्रेट होने से वो रीजन फॉर्म होता है. सो ओनली वन इज इन द बेसिक ग्रुप, बेसिक पीएच, while टू आर इन द एसिडिक रेंज. अब इसमें क्या होगा, उल्टा हो जाएगा. दिस इज फॉर लाइसिन जो कि बेसिक है. इसमें 2 अमाइनो ग्रुप्स हैं जो कि टाइट्रेट हो सकते हैं. सो सबसे पहले मगर कौन सा टाइट्रेट होगा, कार्बोक्सिलिक ग्रुप जो कि सबसे एसिडिक है. देन बेसिक ग्रुप्स एक के बाद एक टाइट्रेट होंगे. ठीक है, तो एक 9 पे जाएगा, एक 10 पे जाएगा, जबकि ये दोनों बेसिक रेंज में होंगे. अब 7 होंगे और बेसिक रेंज में होंगे. यहाँ पे कार्बोक्सिलिक ग्रुप्स तो हैं, तो दोनों कार्बोक्सिलिक ग्रुप्स जो हैं वो एसिडिक रेंज में. यहाँ दोनों अमाइनो ग्रुप जो हैं वो बेसिक रेंज में है. और कार्बोक्सिलिक ग्रुप जो जैसे टाइट्रेट होता था वो दोनों के लिए कॉमन है. This is the repetition. Here they will only have pK1 in acidic range while 2 pK values in basic region, and PR comes after pK2. PR comes after pK2 means pK1 जो acidic range में मिला, pK2 जो basic range में मिला, और pKr जो basic range में मिला, और सबसे ज्यादा या सबसे लास्ट में टाइट्रेट हुआ. Now what will happen for histidine? हिस्टिडीन में कैसे स्पीशीज वेरी करती है. pK1 इसका कहाँ आया, 1.82 पे. अगेन दिस इज फॉर कार्बोक्सिलिक ग्रुप. देन pKr आ रहा है 6. दिस हियर you can see charged unit here. Here this remains as such. Then this species occurs, predominates. Then finally all the charges are neutralized in the process of titration. So pK2 is here, pKr is in between pK1 and pK2, and IPH will be the average of I have shown you the formula. Half means that was the average of all three pKs. तीनों के pK के बीच में कहीं पे IPH होगा, which is isoelectric point, or pH at which this amino acid has neutral charge, and it will not migrate towards any electrode in an electric field. So what is the advantage, or why we are doing this? Each amino acid has its own specific pI, and this varies with the nature of medium also. Okay, if pH of the medium is below pI, then it will, which species will predominate? It will be positive amino acid will be, net charge will be positive, and above pi net charge will be negative. So that depends on the medium in which amino acid is placed. The behavior of a peptide protein in pH of body fluids or in extraction medium depends on the pK values of its constituent amino acids. Table of pKa and pi values of each amino acids are readily available and can be used as standard to identify unknown amino acid. If we know titration curves of all amino acids, we can identify unknown amino acid by titrating that also. Furthermore, identification of a region of titration curve require a thorough knowledge of protonation and deprotonation of an amino acid and an understanding of definition of isoelectric point. आइसोइलेक्ट्रिक पॉइंट को पता होना चाहिए, उसकी डेफिनेशन या उसको उस अगर आई पॉइंट दिया है तो उससे हम मीडियम का पीएच समझ सकते हैं टाइट्रेशन में कहीं पे भी. देन द नंबर ऑफ पीके वैल्यूज differentiates polar and non-polar amino acids from charged amino acids. Position of pKa values for charged amino acid allows one to identify positively charged from negatively charged amino acids. Comparison between experimental and literature pKa values can be allow the identification of specific amino acid. It is possible to predict the ionic species present at pH 1, 2, 3, 4, 5 till 12 in the titration curve with alteration in the charge of an amino acid in the changing pH milieu. It gives quantitative measure of pK of all ionizing groups. Different amino acids can be differentiated from one another on the basis of direction and relative rate of migration of each when a mixture of them is placed in an electric field at a known pH. This was all that we know about titration curves, and if we know the titration curve, we can take advantage of them for identification, separation of amino acids also, and there is net IPH for proteins also, so they migrate towards anode or cathode depending on the pH in which they are suspended, and this is how this titration curve has a very important uh, value to for the protein chemistry. So this was all for titration curve. I hope I am clear about each and every part. Thank you.